couple of quick announcements before we get started. First one, as requested, popular request, we'll have a special CRISPR lecture on Friday by one of my PhD students, George Kaysen, who has been preparing this for his qualifying exam. So he knows way more about it than I do, which is good. And I will make sure that he gets all those slides out to you ASAP. There probably won't be clicker questions that day. Woohoo! But we'll see. If he wants to do it, we'll do them. Which reminds me, clicker question-wise, a couple of things. One of you, don't know who you are, pointed out there was a problem with one of the clicker questions. As you look at the notes, we'll have seen that. I got rid of that. I also went back and checked all of the clicker scores. For some reason, like the last clicker question each time wasn't showing up properly. So I've corrected all of that, and I just tried to upload all the data. I'm not sure if it uploaded properly. So in process of doing that basically as we speak. So are there questions about sort of, clicker things, et cetera? I think there's one or two of you who are still doing your clickers, but I don't have a name for you, I just have a clicker number. So if you think you've been doing a lot of clicker answering and getting a whole bunch of them right, let me know. Just send me your Odin ID and your clicker number, and let me know what's going on with that. Pardon? You've been getting them all right? Okay, yes, you can certainly let me know that that's true, and I'll go back and check, and I'll figure out how many you really did get right. So the first one, again, just this whole clicker business, is here. And that is, dimeric HTH-containing proteins often have their recognition helices about 34 nanometers apart. That's totally wrong. It should have been either 34 angstroms or 3.4 nanometers. I messed up, and I think I misspoke about 2 minutes before I actually gave you the clicker question. So 34 angstroms or 3.4 nanometers just goes to show that I don't proofread these clicker questions as well as I should do, and definitely less well than I do exam questions. And because of this, and I looked at all the answers, none of the answers make sense whatsoever. I just threw that question out. So it's also something I can do with the iClicker software that makes sense there. So today we're going to continue and hopefully actually get through transcriptional regulation. Next time talk about post-transcriptional regulation, and then again CRISPRs, and then the last week a couple of lectures on methods, and then review, and then end of the term already? How did that happen? Any good answers? No, I didn't know how it happened either. So last time we talked about mostly activators and repressors in eukaryotic systems in a very general sense, and also a little bit about chromatin. And then in previous lectures, we talked about DNA binding and bacterial regulators. Today, we're basically going to get into some real examples of how some of this regulation works. And basically, we don't know very much about how this regulation works. It's all a very big picture kind of analysis of this. And one of the papers that some of you may have seen right as you came in that we may get back to and look at later about the glucocorticoid receptor, there's a great quote in there that I'll try and get to when we get that far. But basically, a couple of things about what we do know, what little we do know, about transcriptional regulation in eukaryotic systems. A lot of it has to do with this so-called combinatorial control, and that's multiple different regulators, and those are going to be both the DNA binding regulators, but also the co-regulators that are involved, and then mixing and matching all of these different things. The one example we'll talk about actually is really one of the best defined and relatively simple ones. Then we'll look in a little bit more detail at some of the regulatory circuits, what you can do with various different transcriptional regulators, and then talk a little bit about epigenetics and DNA methylation if we get there. So just the examples we're going to talk about. The first one is positional expression in Drosophila embryos. How many of you have taken developmental biology or are taking it now? Good, because you should have taken this course first. So you won't have heard about this positional expression in Drosophila embryos. This is a really nice example of combinatorial control. Same thing is true of yeast mating type determination. This one has a little extra twist of chromatin remodeling. Switch and lambda, of course, absolutely wonderful system because it's a viral system. But it's a really nice example of how you can have one regulator doing two different things. Eye development, and to some extent I could also put IPS cells here, is a nice example of what are called master regulators. So one particular transcriptional regulator that causes all kinds of changes, just if you express it at the right time, which means that happens properly, or even at the wrong time or wrong place. And then with a little bit of exon activation, this is sort of the flip side of transcriptional activation. This is a really nice example of transcriptional repression and how transcriptional repression works. So one of the first questions that is really uppermost in terms of thinking about particularly multicellular organisms is how do you get all of these different transcription factors, transcriptional regulators, in different cells at different times? Because it turns out that it's really which of those regulators are being expressed when, which is really important in terms of getting particular regulation. And this is just one extremely simple example of how that could work. Just having to do with a simple choice whether the cell after it divides is closer to one end of the organism or the other end of the organism. And because of that, it's going to express one particular regulator or another particular regulator. And it's just this same rule, front or back, left or right, and differential regulation of each of these factors that ends up giving you a whole different selection down here at the bottom of various regulators. And so all it is, each time, if you're on the left-hand side, you're going to get even. If you're on the right-hand side, you're going to get odd. And each time you divide, you get new sets of regulators. So first division, okay, it's just one regulator. So everything that's on the right side has one. That means everything after that has regulator one. Next division, okay, now we've got an even will be on our left side. Odd is on the right. So these 4 cells have different types. These 8 cells have different types, etc. And you can just think about how cells divide and what they'll end up with in terms of these different factors. This is just a very general way of thinking about things. Let's look at a real specific example of this. This is the Drosophila developmental pathways. In fact, this is only one of many, many, many different, extremely well-known processes of looking at gene regulation. This particular one is what's called the EVE gene, or even skipped. And as always with these geneticists, they're going to name their gene based on the mutant phenotype. So in this particular case, if you have a mutation in even skipped, you're going to skip all of the even segments in the fly. So people have then looked at this. It's actually a really nice gene to look at. If you just look at the normal expression of even skipped in a Drosophila embryo, which is what's here on the side. Go back up here. Where's my pointer? Pointer's here somewhere. So it comes on the next slide. There we go. So here's a Drosophila embryo. And if you look at just where even skipped is expressed in the embryo, these 7 literally stripes that go up and down the embryo, in fact, all around it, because this is a 3-dimensional image here. And so these stripes literally just down the embryo from the anterior, the front end, to the back end. But just as important is that in these regions where there's no expression, there's still that gene. It's just a question of why this gene is expressed here and only here in each of these 7 different cases. And it turns out that there are actually 7 different regulatory segments upstream of the promoter in this gene. And you can take one of those regulatory segments and put it next to a reporter gene, our friend LAC-Z, the beta-galactosidase gene, enzymatic activity that Drosophila normally doesn't have. And if you express this gene and stain these cells with a dye that requires that enzymatic activity, you'll find that it's expressed in only one place. And that's just due to this particular regulatory element right here. So how then does the embryo know, again, totally over-anthropomorphizing here, to express this gene based on these sequences. Turns out it's due to the presence of 4 different proteins: bicoid, hunchback, giant, and cripple. Again, the Drosophila geneticists are naming all of these genes based on their mutant phenotype. And we can take a quick look at them in just a second. But Ian had a question first.
He does have a question. So when you say even skipped, unless this isn't important, when you say even skipped, does that mean it's going to skip stripe 2 regulatory and stripe 4 and stripe 6?
OK, so even skipped. OK, unfortunately, so your question is, what's the phenotype of even skipped? And does it even make any sense here as far as this is concerned? The answer is actually no. It has nothing to do with any of these stripes at all. It has to do with other parts of the embryo. So it's a downstream effect of lacking this particular gene. Does that make sense? Yes. OK. So the expression of even skipped, before you get on to that next step, has to do with 4 genes: bicoid, hunchback, giant, and cripple. How many of you speak or understand some German? A few of you. So a lot of the original Drosophila geneticists, in fact, some of the very best Drosophila geneticists, are Germans, and so they come up with these names for their mutants. So this particular one was crippled as a cripple. Giant, what does that mean? If you knock out this gene, you have giant embryos. Hunchback, you knock out that gene, the embryos are crunched up. And so that's where these names actually come from. Unfortunately, they're not terribly helpful as far as this is concerned. What's much more important is that 2 of these are repressors of transcription, and 2 of them are activators of transcription. So cripple and giant are repressors of transcription. Bicoid and hunchback are activators of transcription. And here, this is a reminder, if you look at where these particular regulators are expressed in the embryo, they also have this sort of either striped or differential location in the fly embryo. So the front end of the fly embryo, so the point on it, it's the anterior end, it's the posterior end. Both bicoid and hunchback have these gradients. That means they've got high concentrations at the anterior end and go lower as it moves posteriorly. Giant is present in stripes, actually regulated in a very similar way to even skipped. And cripple also as well has this stripe. One other thing that I forgot to mention here, these embryos are really neat systems because they've got a whole bunch of nuclei, so they spread all the way around the outside of the embryo, but it's one big cell. And so any particular regulator is going to be interacting with whatever nuclei are there. You don't have to have nuclear transport getting from cell to cell, et cetera. So it's a really nice system to look at. OK, so now we have these 4 regulators, 2 repressors, and 2 activators. And then we have the sequence up here in gray of that regulatory region. So the regulatory region, again, you just need this piece. It gives you expression of whatever is downstream of that in stripe 2 and only stripe 2 in the embryo. And that's due to all of these 4 different proteins. So through the methods that we talked about last time, DNA binding, what the sequences are, footprinting, gel shift, etc., we know that the repressors bind at these various different sites on the DNA, and the activators bind at these various different sites on the DNA. Also, what we know is you only will get expression driven by this piece of DNA in the presence of all the activators, but the absence of all of the repressors. So how does that give you a particular stripe? Well, if you remember from here, we've got gradients starting at the anterior end of bicoid and hunchback. These are your activators. And then differential expression of giant and cripple. And what that means, if you just go from one end of the embryo to the other end of the embryo, just looking at the concentrations here, you've got hunchback that then goes down, bicoid concentration goes down, giant and cripple have much sharper definitions. So here, got giant up until stripe 2, cripple right after stripe 2, And what it means is if you look at this piece of DNA, this is the only position in the embryo where all of the activators are present and none of the repressors are there. Anytime you start to have repressors, then they're not going to be allowing the binding of these activators. It's true on this side, you've got giant around. On this side, you have cripple around. So the only place in the embryo that this gene segment will allow you to get transcription, even though this gene segment shows it's present all the way across the embryo. Every single one of those nuclei has this particular piece. You're only getting expression from that particular piece in this particular region in the embryo. So 2 repressors, 2 activators, have to have all the activators bound, can't have any of the repressors bound. Because of where they are in the cell, that gives you expression in this one particular position. And again, this is a ridiculously simple version of most gene regulation. Questions? Yes?
Are these transcriptional regulators, are they specific to stripe 2 and none of the other stripes?
So the question is, are these particular transcriptional regulators just specific to stripe 2? In this particular set, combination of factors, which is important for stripe 2. There are multiple other combinations which are going to be important for the other 7 stripes, or the other 6 stripes, I should say. Some cases, not surprisingly, anywhere which is anterior here, you'll be using bicoid and hunchback as regulators, because they're activators of expression. However, if you're going to think about some of the segments down here, some of the stripes over here, you can't be using bicoid and hunchback in terms of activation because there isn't any there. So yes, some, but certainly not all. Okay, other questions? Good, everyone's got their clickers out. Perfect, well-timed. You might want to be looking at your notes for this too. If you're investigating Eve expression in a Drosophila embryo that's missing cripple, you would expect normal Eve expression lacking in stripe 2, but normal in all other stripes, additional Eve expression posterior to stripe 2, additional Eve expression anterior to stripe 2, or no Eve expression whatsoever. Find somebody who's got a copy of the notes. This will help. Sounds like we're going to want more time. Is that true? Yeah. I'll start it again just a sec. Your second round. And yeah, be sure to vote now this time. That first set of votes is going to disappear. It works. It's the last one that you push that counts. Ten. 4, 2, 1. Okay, what do we think? We have a difference of opinion here. But for the most part, people got it right. A Eve expression posterior. And why is that? Cripple is posterior, and there's still a little bit of those transcriptional activators there. And if you look at the graph of where they are. So normal Eve expression you definitely wouldn't have because you're missing that one particular regulator. Lacking in stripe 2 but normal in all other stripes, no, because you're missing a repressor. Posterior, again, because yes, you've got the activators, but you're missing the repressor there. Anterior, posterior, front, back. If I'd said something about giant, what would the answer have been? It would have been D, and no Eve expression, because we had this great question here. What about the other stripes? Most of those are going to have nothing to do with cripple whatsoever. So the correct answer is C.
Is there any way that B would be the correct answer?
So completely lacking in that stripe, again, that's going to depend on exactly what the other regulators are that are being used. And since anterior to that, you probably have activation by bicoid and hunchback, it's unlikely. OK, so that was it for combinatorial control, at least for now. We'll get back to another example of that in just a second. I wanted to talk about some master regulators. Again, these are these crazy fruit fly geneticists who've come up with some of these things. Having a compost bin, I think, means you know a lot about fruit flies. At least you see a lot of fruit flies. So really, really, really easy to breed, and the genetics have been extremely well worked out for these organisms. One of these that's been really quite well studied are some genes that are important for development, particularly development of individual structures that you find in the fruit fly. And one of the best and easiest, actually, to look at are eyes. They've got these really amazing, red eyes for the wild type. And anytime you make mutations in that, it's really easy to see when you're just looking at the fruit fly is there's something weird about that particular eye. And so one of the genes that was found very early on was a gene called, you guessed it, eyeless. So what happens when you mutate this gene? It doesn't have an eye. But one of the other tricks that a lot of these geneticists figured out is you can now take these genes and express them somewhere else in the fly. And in this particular case, what they did is they expressed eyeless in a part of the embryo which gives rise to legs. And it doesn't develop actually all the nervous structure. They can't see out of their legs. But they have this whole development of the eye structure that ends up on the legs. Another one, which is a really famous particular mutation, is called antennapedia, which is instead of having legs, they have antenna at each of these. So just really crazy flies. And again, back in graduate school, to make the connection to my PhD thesis, there are a number of fruit fly genetics labs working in the same building, and we'll talk about some of the actual mutants that they were working on Wednesday. But we would have our happy hour after seminars where there was fruit bowls and things lying out, and some of my colleagues go, "Oh, there's my curly-winged sex-combs reduced mutant." And so, yeah, lots of different fruit fly mutants around that are there. So, but it's just expression of this one gene that will then lead to expression of lots of other ones, which will eventually lead to the expression, in this case, of eyes. Probably the most extreme examples of these are the ones that I've been talking about quite a bit. These are the amazing pluripotency genes, originally found as 4, and that's what the group got the Nobel Prize for. Now it's down to 3. 3 transcriptional regulator proteins, OCT4, SOX4, and KIF4, which you can take and put into the nucleus of a cell. The way you do this, of course, is with viruses, because they're the best way of doing anything. And then the expression, and particularly the overexpression of these genes, gives you what's called an induced pluripotent stem cell. The induction is because of these 3 genes being expressed here. With this induced pluripotent stem cell, if you give it the right signals, usually where it is in a cell, other external factors, etc., these can develop into pretty much any different cell type, and in a few cases, actually, a whole organism can be derived now from this fibroblast. And so this is a really amazing technique, because when we talked about the beginning of the discussion of gene regulation with the dollies, et cetera, you had to have embryos and then take those embryos and put a new nucleus into them. Well, now you can actually take a cell, no longer needing embryos, and convert it into a pluripotent stem cell that then you can use for all of these different mechanisms. So hopefully, although there are exceptions to this, should be able to completely avoid needing embryos for a lot of these kinds of experiments, or embryonic stem cells. So a lot of the work that's going on in stem cell biology now has to do with these iPS cells and exactly how you can get them regulated. There's some issues because you put these in with viruses. What do you do with these extra viruses that are in here, which are also in these cells, et cetera? So there's some, it's not a perfect system by any stretch of the imagination, but it's a really, really, really good one. And again, these are the sort of master regulators, a lot like the ILIS protein, which will then regulate multiple other transcriptional regulators to give you one of these particular states here. So an overview of this system is really like this, which is sort of a ridiculously complicated system that who the heck knows what's going on, so how the heck can Stedman ask us questions about it? So really looking at each of these individual segments of your DNA, all of the proteins that are associated with that, either directly DNA binding proteins or indirectly these co-regulators, there's always going to be sort of a mixture of activating, repressing, activating, etc. And all of that is going to lead to a particular probability at any given point of getting gene expression to take place. And because of this whole complication business, it makes it ridiculously hard to predict what kinds of outcomes you're going to have given a particular stretch of DNA and a particular promoter. And probably the best studied of these is a protein called the glucocorticoid receptor. And the glucocorticoid receptor is, again, one of the best studied of these gene regulatory proteins. Why? Well, because it's there to some extent. It looks like it's present in pretty much all vertebrate cells. There are few exceptions, but for the most part, it's actually there. It's normally present in the cytoplasm, so outside the nucleus, and so because of that, it's not active. It's outside the nucleus. All of its activity, so all the DNA is present in the nucleus. The other thing that was found is that in the absence of some kind of ligand, this red thing here, it stays monomeric and it stays out in the cytoplasm. So it's only in response to a particular signal that this glucocorticoid receptor comes into the nucleus and then has some kind of activity. What is the classic ligand for this? Cortisol, pretty much any steroid hormones, and interestingly enough, RU46. Everyone know RU46? Yes? Good? No? Heh? RU46, the abortion pill. What's amazing is that it works. It also makes you rather sick in the process, at least what I've been told. Haven't taken it myself. For hopefully obvious reasons. This interacts with the glucocorticoid receptor as well as the progesterone receptor, which is the main target of that particular drug. So these steroid hormones will bind to the glucocorticoid receptor, get it to go inside the nucleus. And the classic example again here is cortisol. What happens? Stress, cortisol levels go up. Physiology. How many of you taking physiology? Good, so you know more about this than I do. The stress level goes up, cortisol levels, a bunch of these steroids will go up, and then one of the things we have to do is get a lot of energy. Stressed out about this. And so usually that particular aspect of cortisol signaling has to do with turning on metabolic genes in the liver, multiple different metabolic genes. But it turns out that this particular response, the stress response, goes all over the body and does lots of different things in lots of different places. And a lot of that is going to depend on who the partners are for the receptor once it gets inside the nucleus, again, in the presence of this drug actually being bound to it. So here are just 3 examples of different regulator proteins that are bound to the DNA and end up being stimulated, in this case, by the presence of the glucocorticoid receptor. And this is, again, a nice time to go back and take a quick look at this paper, which gives you more information than you'd ever want to know about it. But what I really wanted to pull out of this paper was one quote, which is, where is that? It's a little bit later on here. Here we are. So these guys are all acting, as we've talked about before, a lot of these transcriptional regulators, thousands of nucleotides. In the case of the glucocorticoid receptor, up to 10,000 nucleotides away. And this is the quote here: such action at a distance is well documented, heavily studied, and essentially not understood. So that gives you a bit of an idea where we stand with a lot of these regulators. It's actually a really, really nice paper in terms of how this particular transcriptional regulator is working. Again, probably one of the best studied of the transcriptional regulators, and again, practically not understood exactly how it works. But their sort of take-home message from the paper is that the glucocorticoid receptor by itself is really kind of serving as a scaffold for getting lots of other things to associate with it. And it turns out that there are thousands of other things that are going to be associated with it. And so little changes in terms of exactly what steroid hormone it's interacting with, if it's cortisol or RU46, exactly what DNA sequence it's binding to, exactly who its partners are, all of that is going to determine what the actual outcome is of this steroid hormone receptor binding to DNA and its ligand in terms of the actual output. So that's the glucocorticoid hormone everywhere, but it's really dependent on context. And then the next example is mating type switching. I apologize here. This is one of the pieces that got taken out of this version of the textbook, but the previous version of the textbook actually have them in it. So you can go to NCBI, and I'll try and get some of the additional pages on this a little bit later on. Yeasts are a wonderful system for studying eukaryotic biology because they're very small. You can make lots and lots of them. You can also use them to make beer. But one of the really fascinating things about yeast is that they can, at least the Saccharomyces cerevisiae, they can exist in 2 different states, basically sort of the male and female equivalent, only they're A's and alphas. And these particular organisms are probably a lot better off than we are because they can switch from A to alpha and alpha to A. So how that process actually works is a really fascinating process, and we'll talk about that in a little bit more detail. What it means is that A's and alphas just in and of themselves can replicate as haploid organisms, but you can also have A's and alphas come together and make a diploid organism so you can have sexual reproduction. So it's a really nice system if you want to study haploid replication or if you want to study diploid replication and looking at the various different proteins. There's only about 6,000 of them in yeast are involved in this whole process. So how do you do mating type switching? And this is again from the 5th edition of the textbook here. So you can go from alpha to A and A to alpha. How does that happen? Well, it turns out that all yeast cells have a copy of the alpha information and a copy of the A information, because where else would you get all this information from? That's present in the genome in a silent state. And we'll look at that silencing in just a second here. So alpha silenced, A silenced. What's important for how the cell is reacting is what genes are being expressed. So is it the alpha genes that are being expressed or the A genes that are being expressed? And what's called the mating type locus. So that's an active part of the genome. So how do you change? How do you change from A to alpha and alpha to A? Turns out that this is a really nice example of basically homologous recombination and repair of double-stranded breaks. Here, where does that double-stranded break come from? It comes from an endonuclease. So an endonuclease will literally cut in this locus, and then it will get repaired. And what's interesting is it always gets repaired from the opposite one. So if you have an alpha that gets cut, it gets repaired by A. If you have an A that gets cut, it gets repaired by alpha. So it's basically almost identical to the DNA. There's a repair mechanism for double-stranded breaks, is this mating type switching process. Next question is, how do you have these silenced genes? Yeah?
I'm sorry, you said if A is cut, repaired by alpha, and if alpha is cut, A is...
It's repaired by A, yes. So it always switches, goes from one to the other and back and forth. And that's actually also shown here. Excuse me, come on, let's go backwards. The switching, computer's not talking to me here. Switching takes place with this activity of the endonuclease. The endonuclease is also called HO for reasons that we're not going to get into here. If you're interested, ask me on email. I'll let you know. But HO is the endonuclease. So if the endonuclease is on, you have to switch from alpha to A, on again, A to alpha, etc. If it's off, you always stay being one of these. So that gives you the change, but how do you have these other two sets of information that are not being expressed? Well, surprise, surprise, it has to do with heterochromatin. So another review of what we've talked about already. Heterochromatin for the alpha and the A, they're stuffed off down at the ends of chromosomes, quite close to the telomere, but most importantly have a silencer which is right next to them. Now, one of the things that we've talked about before has to do with chromatin remodeling complexes. Anyone remember what some of those chromatin remodeling complexes are called? A classic example of those is something called SWI-SNF. Where the heck does SWI-SNF come from? What are we talking about here? Mating type switching. I wonder if SWI has anything to do with mating type switching. And if you have a SWI mutant, it's going to have trouble with chromatin remodeling. SWI mutant means it doesn't switch. It means you're not remodeling the chromatin. So chromatin remodeling is absolutely critical for this switching step. From silenced due to heterochromatin, to being moved over and being put into the active region here. So the last thing I want to talk about in terms of yeast mating type has to do with what's actually being expressed here. So we've always got two copies, two silenced copies, A and alpha. It's what's actually being expressed at the mating type locus, the active part, which determines whether you're an A cell or an alpha cell. But also, again, the wonders of yeast genetics here, turns out that if you have a diploid cell, the only way you can make a diploid cell is if A and alpha come together. And that's the only way you can end up with diploid. And they make these wonderful things they call shmoos. So you got a shmoo, and this is the actual genetic description of this. It's the fully scientific name is a shmoo. When you get an A cell and an alpha cell right next to each other, they shmoo. And you have the fusion, the diploid, which takes place. This is a way that you can tell if you've got an A or an alpha. Otherwise, it's really hard to tell if you've got an A or an alpha. Are they forming shmoos or not? So what happens in this case? Not surprisingly, and partly because we're talking about this in the transcriptional regulation section, the A locus is expressing a transcriptional regulator, and the alpha locus is expressing two transcriptional regulators. So if you have an A cell, it's expressing A1. Turns out that A1 just by itself doesn't do squat. You have your A-specific genes that are turned on by a cellular factor, which is present in all cells, in A cells and alpha cells, etc. So that turns on all of your A-specific genes. It's these ASGs. Alpha-specific genes are off. Haploid-specific genes are on because you don't have any kind of regulator that's dealing with this. So again, MAT-A1 just by itself does nothing. We'll get back to MAT-A1 a little later on. Alpha, on the other hand, has two regulatory proteins, alpha 1 and alpha 2, again, ridiculously creatively named here. Alpha 2 is a co-regulator of this general protein here, which now turns off the A-specific genes if you're an alpha. MAT alpha 1 is a co-regulator that brings MCM1 to the alpha-specific genes. So MAT A1, MAT alpha 1, MAT alpha 2 gives you in your haploid cells A, because A1 does nothing, alpha, repressing A-specific and stimulating alpha-specific genes. The last case is once you have your shmoo and you form a diploid, now you have MAT A1 and MAT alpha 2. MAT alpha 1 is actually repressed in these cells. Turns out it has a little bit to do with MAT A1, but we're not going to get into it. Because you've got MAT alpha 2, you shut off the A-specific genes. Because you don't have MAT alpha 1, you don't have alpha-specific genes, but now you turn off your haploid-specific genes. And your haploid-specific genes are what you need to shmoo. Because if you bring two diploids together, you don't need to shmoo anymore. So that's, and there are multiple other genes as well. So it's just three genes that are going to control all of your A-specific genes, your alpha-specific genes, and your haploid-specific genes in combination with one cellular gene. What? Confused? Confused enough that we can answer a clicker question on it? No? It's Stedman logic. You know how logical that is. So, now let's actually stop this and have a question first. That might help. In the absence of MAT alpha 1 in a diploid cell, you would expect expression of haploid-specific genes, expression of alpha-specific genes, expression of A-specific genes, A and B or A and C. Pardon? Oh, it's MAT A1, sorry. What I write, not what I say. What I do, not what I tweet. Looks like we might want another round for this. Yes? Okay, we can do that. See, I can see that there are only 50-odd of you who've actually voted here, so. Okay, round two. Ten. Okay, I'm actually going to go back one here before we look at the results. Oh. So here, actually maybe go forward one. Expression of A-specific genes is or haploid alpha-specific. So if we go here, now I'm going to move this, try and move this out of the way now. be a little bit easier to look at here. So we're missing MAT-A1. So that means, what about haploid-specific genes? Haploid-specific genes be on. They're always going to be on in the absence of MAT-A1. Okay, so we know that we're going to have A. What about alpha-specific and A-specific genes? And that's why we actually need to get rid of this here. We'll, if my computer, say the technology here is not working quite as nicely as I would like here. But so what about alpha-specific genes or A-specific genes? A-specific genes are going to be expressed now with MCM1, just like we have up here at the top. A-specific genes are going to get repressed by MAT-alpha-2, right? And in a diploid cell, we've got MAT-alpha-2. What about alpha-specific genes? Alpha-specific genes are going to get turned on by MAT-alpha-1 and MCM. So what does that mean about our diploid cell? MAT-alpha-1, we're not going to have MAT-alpha-1, so we're not going to be turning on alpha-specific genes. We're not going to have A-specific genes because those are turned off. What does that leave us with? Just our haploid-specific genes. Okay, yes, no, yeah. Pardon?
So it's A. On the question, did you say it was the absence of the MAT-A gene, not the MAT-alpha gene?
Yeah, but that's you don't have the MAT-alpha-1 in diploid cells.
That makes no sense. But
Yeah. Pardon? Why does MAT-alpha-1 get repressed? It actually has to do with the, it's not just MAT-A1. There are other repression things, so you don't have it. It's diploid-specific genes. So maybe we'll have to throw out that clicker question. Oh. Okay, let's forget about all this boring eukaryotic stuff. Let's talk about interesting stuff, like viruses. So part of the reason to study viral regulation has to do with the fact that they're nice and simple. So the case of lambda, we already talked about the lambda quote-unquote repressor. So lambda is a really wonderful system because it's got two sort of choices. You can either, when you infect, sorry again to totally over-anthropomorphize here, you have an infection with your bacterial virus that can just make a whole bunch more virus. And this is called the lytic state. This is great if there are a whole bunch of host cells around. You can go and infect those and then make more virus and infect more cells, etc. If, on the other hand, and this is true probably actually for most viruses, there aren't very many of your hosts around and you're completely dependent on a host, then you probably want to hang out inside that host for as long as you possibly can. And that's what's called the lysogenic state. And we talked about the integrase before. We talked about site-specific recombination. Takes the viral genome, puts it into the host genome, and you can hang out for basically as long as you like. So this is the state right here, where you have the lambda repressor protein, which is serving, again, confusingly, unfortunately, as a transcriptional activator. It's expressing its own gene. So lambda repressor leads to expression of lambda repressor. This is called the C1 gene. We'll talk more about the C1 gene later on next term. On the other hand, so this is what's happening in your lysogenic state. You're just hanging out. You're making a whole bunch of lambda C1. That bunch of lambda C1 shuts down the expression of your other genes that you need to make more virus, but is making more and more of this regulator. On the other hand, if there's a bunch of good, happy cells around. Then you want to make a bunch of virus, and then you make this other protein called the control of regulator and other proteins. We actually looked at this when we looked at our dimerization earlier on. This binds to exactly the same place as where the lambda repressor binds and actually blocks lambda repressor binding. And now you have activation of all of these other genes, including lambda cro. And so it's this switch between, are we making lambda repressor? Are we making lambda cro? Once you've made that decision, everything else, because of these positive feedback loops, is going to stay that way. So make your switch, make your decision, always go this way, always go this way. Now, of course, it's how that switch decision is made. Come back next term for virology, and we'll talk about how that switch is. But the important thing here is these are two regulators which are in positive feedback loops. And those positive feedback loops give you what's also called, and the textbook gets way into this, cellular memory. So it says a decision that you've made, you remember that decision. And how are you remembering that decision? It's because of this positive feedback loop. And so in a much more general sense is this. So there's some kind of signal that happens, turns on the expression of A. A then expresses more A. When you have cell division, each of the daughter cells gets a little bit of A, which turns on expression of more A. These divide, get more A, etc. This is just one of the regulatory circuits that you can think about in terms of gene regulators. This is an activator turning on an activator. So this is a positive feedback loop. Yeah?
On the previous slide,
Mm-hmm.
it looked as if the lambda repressor is turning off the CRO, which
Mhm.
is after it, which
Mhm.
makes sense. But the operator is turning off the C1, which is before it. Thank you. This
Okay.
It's transcribing along. It's coming to the C1 before it hits the operator, and then is transcribing the CRO aspect of the gene. How is it turning off the C1, which is upstream from the operator?
Okay, so the question here is, I'm going to paraphrase and tell me if I'm getting it right or not here. So how can the operator be working on two different genes? And the answer is it's just where the polymerase is going to interact. So this is a great review thinking about gene regulation in bacteria. It's where exactly the polymerase is going to bind relative to these other binding sites. And they try and show it here. You've got these guys binding on this side of the operator and this guy binding on this end of the operator. So this is going to block binding by the polymerase going this way. It's going to allow binding by the polymerase going that way. Here, this guy blocks the polymerase going this way and stimulates the polymerase going that way. So it's one operator that's working on two genes. Okay, so positive feedback loops. Positive feedback loops are only one of the possibilities that you can have in terms of these gene regulatory circuits. The easiest one to understand. Yeah, Ian.
So one operator can be responsible for the expression of two operons?
So the question is, can one operator be responsible for the expression of two operons? Most definitely, yes. Yeah, because this is DNA sequence. That's all it is. Okay, so positive feedback loops are probably the easiest to understand of these possibilities. You can also have a negative feedback loop, and negative feedback loops are just a transcriptional regulator now represses its own transcription. So if you have some signal that says turn on a repressor, it'll get to a certain level and then it will stop because it'll be repressing its own regulation. And so if you think about, again, the whole case of the lambda repressor, it's stimulating through the positive feedback loop its own expression, but it's shutting down anything else. If you have the repressor and just a repressor that represses its own, you'll have a signal, it'll be on, turns on the repressor, and then just damps that signal completely. And you can set up all kinds of different regulatory sequences this repressor represses this repressor, which represses that repressor, etc. There are lots of different circuits that you can get into here. This is one of the things, I think didn't bring my copy of the textbook. On the front cover, it's got all of these arrows going for activating, repressing, etc. That's what people are trying to do in terms of what they call systems biology, trying to understand through each of these different interactions what's leading to expression of one gene versus another. And another one to look at here is the so-called flip-flop switch. That's just repressors leading to repressors. The last one that I wanted to talk about here, we'll talk about those repressors in just a second here, is the so-called feed-forward process. So we have some kind of input, gene regulator that regulates a second gene regulator, and then you have some kind of output, but it's dependent on both of these regulators. So basically what this does is it says if you just have a blip, some kind of little input, some kind of random fluctuation, which is what we always see in biology, is random fluctuation. If you just have a little blip, A is turned on, but there's not been enough time for B to get turned on in terms of finally having your output here. If, however, you have a signal that goes on for a long time, that tiger is getting closer and closer to you, so you turn the cortisol up. So then you do want to have a response. And so that's what happens here. If you have this long enough input, you've got enough time for B to be expressed based on A, and A is still going to be around, so now you finally have an output. And it's this combination of positive feedback, your cellular memory, negative feedback for keeping things at a nice level, flip-flop, feed-forward, and then this last case in terms of these regulatory sequences is this repressor process. And so keeping things at the same level, but actually you can use these kinds of repressor circuits to give you a oscillation pattern which happens. And again, it just has to do with the fact that transcriptional regulation, it's turning on making your RNA. That RNA gets translated into protein, and if you're thinking about this in a eukaryotic cell, that protein is being modified, folded, etc. So there's always going to be a time delay between the expression of one protein and, the transcription, I should say, of one gene and the expression of that particular protein. And so this is a process here, which it turns out is used for circadian rhythms for that seminar that we didn't get last Thursday, which I'm bummed about. So expression of a repressor will allow you to have expression for a little while, but then shut it down. And then once that's been shut down, you'll have expression of the next gene, which will shut down expression of the next gene, which will shut down expression of the next gene. So in theory, just starting out here, we have expression of A, which will then get blocked by expression of C, which will allow each of these proteins to be expressed really very much in a circuit. And this was predicted, was actually done. That's what this figure is down here at the bottom. And you can literally design these kinds of processes. And again, it's exactly what the synthetic biologists are trying to do, is design genetic circuits based on what we know about each of these individual proteins. Yeah?
So would the repressors have another function here other than to just repress that site? I mean, what would be the purpose of just expressing a loop of proteins that just turn each other off?
Why do this, basically? So it turns out that circadian rhythms happen exactly this way. Now, one of the things which is really important about that, if you have this protein that's around all the time, then you're not going to get this oscillation. You have to have some way of degrading each of those proteins so that each of them has a particular half-life. And so if you do that, you can literally set up a rhythm. And partly why we get jet lag has to do with cycles that happen in an extremely similar mechanism to this. So all organisms have a circadian rhythm set to about 24 hours. And these kinds of repressor circuits and degradation of the repressor each time gives us that kind of cycling. Really pretty amazing process. Now I cut out some of the, if you look at some of the other slides, we talked about the clock genes and so on and so forth. We're not going to talk about them this term. So I want to finish up with X inactivation. As I've mentioned, I think many times, half of us here are genetically superior. I'm one of the inferior ones because we've got, I've only got one X chromosome. But there is a problem with having two X chromosomes, and that is we have to deal with males who only have one. And so expression from the X chromosome has to be, in most cases, a few little exceptions of individual genes, you just have to have expression from one of the X chromosomes. If you had expression from both of the X chromosomes, you have a lot of trouble. So this is a process called dosage compensation, and it happens in other genes as well, but for the most part, it's best understood what goes on in the X chromosome. So in mammals, this is what happens. You have one of the two X chromosomes that gets inactivated early on in development, but not immediately after your embryogenesis. Once those get inactivated, it has to do with a heterochromatin formation that compresses the chromosome and basically inactivates almost all of the genes. And there are a few exceptions to that. The book talks about the calico cat example, where female calico cats will have mixed colors, and that's because the coloration genes are on the various different X chromosomes, which one's inactivated and which one's not. We know quite a lot about how this actually inactivation takes place, and this is one of the really nice examples of it's all about the RNA. Actually, it has very little to do with the protein. The so-called X inactivation center expresses a long non-coding RNA, more than 200 nucleotides. And what that does is, after expression, it coats one of the two chromosomes and is basically moving along that chromosome. After you have that, you have removal of acetyl groups, so condenses your chromatin. The histones get methylated. You put on an alternative histone, and DNA methylation takes place. It's the combination of really these four things: taking acetyl groups off, methylating your histones, putting the alternative histone, and then finally methylating the DNA, which really gives you that massive compaction which shuts off expression of all the genes on one of those two X chromosomes. I think, for want of time, we'll probably talk about DNA methylation next time. Yes? Good. Wednesday, DNA methylation. See you then.